So dear students, so today our topic is biosynthesis of pyrimidine ribonucleotides. So already we learned regarding the biosynthesis of purine derivatives or purine nucleotides. So purines and pyrimidines both are required for the synthesis of DNA and RNA, and both DNA and RNA, so they are play an important role in the regulations of the various biochemical process through a involving the biosynthesis of the proteins. So now we first go for regarding the, that is a pyrimidine nucleotides. So biosynthesis of pyrimidine nucleotides, so that can occurs by a de novo pathway, so as well as the salvage pathway. So like the purines, so here the pyrimidines also, so they are synthesized by the two pathway. One is the de novo pathway, so as well as the salvage pathway. So de novo pathway, so they are utilize the amino acids, so they are utilize the certain components and involved in the biosynthesis of the pyrimidine bases first. From the pyrimidine bases, so they are involved in the synthesis of, that is the pyrimidine nucleotides like a cytosine, thiamines, and as well as the uracils. So that pathway is known as the de novo pathway. And regarding the salvage pathway, certain intermediate metabolites, so they are present or formed in our body. Okay, so due to metabolism of, once again, pyrimidine nucleotides are certain other compounds or biomolecules, certain intermediate compound directly available for the biosynthesis of the pyrimidine nucleotides, and such pathway was known as the salvage pathway. So salvage pathway, one of the beneficial pathway, because so that was in avoid the excess of catabolism of, that is the pyrimidine nucleotides. So after biosynthesis, after functions, so they are catabolized also. So due to catabolize, so the more waste material can be formed in our body. So that waste material regarding the uric acids or many others components are formed by catabolism of purine, so as well as the pyrimidine bases. Okay, so those elevated level was responsible for to cause of the various disorders related to the CNS or other body systems. But so due to presence of the salvage pathway, so those intermediate, so they are utilized, or they are, those are metabolites, so they are utilized, then they decrease the chance of the development of the diseases related to the pyrimidine and purine nucleotides catabolism. So that's why the salvage pathway is the minor pathway. Still, it is most important pathway, so that can avoid the certain disorders. So regarding the pyrimidines, so these are the pyrimidines, that is cytosine, thiamine, so it is only found in the DNA, and as well as the uracil, it was only present in the RNA. This is the only one difference in the pyrimidine bases in the RNA as well as in the DNA. So the pyrimidine synthesis process, that was similar to the purines, but it is not a complicated that much of the purines. The pathway is same regarding the de novo pathway and as well as salvage pathway. So both pathway are present in the synthesis of purine as well as the pyrimidines, and even certain synthesis of the basic molecule. So in the purine biosynthesis, we require the first IMP, that is the inosine monophosphate. That is the basic molecule. And here also some basic molecule is required. Further synthesis of the pyrimidine nucleotides. But the process is a not complicated as compared to the synthesis of the, that is the purines derivatives or purine nucleotides. It is a less consuming, that is the process or reactions or number of reactions will be the less, or components will be less required for biosynthesis of the pyrimidine basic molecules. So here also, like a purines, in the de novo synthesis of the pyrimidine bases, the first, the pyrimidine nuclei is synthesized. So after synthesis of the basic molecule for the pyrimidines, so later on, so they are attached to the ribose phosphate. Then so they are responsible for the formations of the pyrimidine nucleotide. So like the same process, like the purines also, the first, the basic purine molecule is synthesized. From the purine molecule, basic molecule, the further adenine guanine is synthesized. So like that only. So in the pyrimidine biosynthesis, first the pyrimidine bases is synthesized. From the pyrimidine bases, cytosine, thymines and uracil, so they are synthesized. Okay, after that, so they are attached with the ribose phosphate, then so they are responsible for the synthesis of the, so these pyrimidine molecules. So now we can learn regarding the significance of the pyrimidine synthesis. So pyrimidine nuclei or nucleotides, they are common with the purine nucleotides. Those are required for the synthesis of DNA and RNA. So pyrimidines, so like a purine nucleotides, so these are required for the synthesis of DNA and RNA. And DNA, RNA, so they are play an important the protein synthesis as well as transfer of the genetic informations from parents to the offspring. That is also role of the DNA and RNA. Even the DNA and RNA from the microorganisms, so they are involved in the to cause of the disease or progress of the disease or growth of the disease. If your molecule, if your drug, any kind, so that if that was inhibit the that is the synthesis of DNA and RNA by inhibiting purine nucleotide or by inhibiting pyrene nucleotide synthesis or directly, so they can denature the DNA and RNA, means definitely, so that molecule or drug used as a drug that was helpful for treatment of the cancer or helpful for treatment of inflammations or any bacterial infection, viral infection, then as well as fungal infections. So that is only by breaking or inhibiting the synthesis of the DNA and RNA. Okay, so here, so these pyrimidine nucleotides also participate in the intermediate metabolisms. Like a purine, the pyrimidine nucleotides are also involved in the biosynthesis of glycogen and as well as the biosynthesis of the phospholipids. So like the purine, so these are also involved in the biosynthesis of the glycogen and as well as the biosynthesis of the phospholipids. And pyrimidine is also having the variety or many biological activities, and is used as a drug in the treatment of the many disorders. So main, the pyrimidine, so they have the biological activities like they are used as the antimicrobial agents. So pyrimidine derivatives, not a cytosine, thymine, uracils, so their derivatives are those drug, so they are synthesized from the cytosine. Structural modifications is there. So due to structural modification, so they look like a cytosine, but they are not a cytosines. So their analog is a cytosine, but they are not a cytosines. So that's why, so they are incorporated the cytosine in DNA and RNA synthesis in the microorganisms. So due to that, the DNA and RNA is not completed synthesis in the microorganism. So due to that, the microorganism growth can be stunted. So due to that reason, so whatever the pyrimidines derivatives are there, so they are helpful for, are used as a antimicrobial agents. Okay, their basic molecule is pyrimidine, or they are synthesized from the pyrimidine basic molecule by structural modifications. So that structural modifications you should be learned from the medicinal chemistry or molecular biology of the that drug or disease. So in the drug or disease, so that where you can learn, so due to additions of certain group in that molecule, suppose the certain group on the pyrimidine molecule, so that can helpful for to kills the microorganisms. So that molecule only they are added. Okay, or for example, if you know that that is regarding the COVID RNA, so that may be the RNA derived viruses or DNA derived viruses. So first they are concluded whether it is the RNA or DNA. Then so they are planned to incorporate the sequence of the RNA in the COVID vaccines, that is the COVID viruses. Then they successfully made, means definitely they inhibit the multiplications of the viruses. Then that is known as the drugs. Okay, so that's why, so these pyrimidine derivatives, so they are mainly used as a antimicrobial agent and also help used as the CNS depressant. So that's why they are helpful for the treatment of the convulsions, helpful for the treatment of the many CNS disorders, or also they can treat the sleeping problems. So they induce the sleep. Okay, so then they are helpful for treatment of inflammation because they are also having the anti-inflammatory activity, analgesic activity, anti-convulsant activity, helpful for treatment of the epilepsy or convulsions, and also they are helpful for treatment of the cancer. That's why they are having anti-cancer activity and anti-emetic and antioxidant even. So they are the herbicidals. So these are the various biological activities. So those are related to the pyrimidine nucleotides. Okay, so their derivatives. So they will be having the diverse biological activities. So they are helpful for treatment of the various disorders. So regarding the de novo pyrimidine synthesis, that is nothing but the synthesis of the pyrimidine bases by utilizations of the amino acids are involved in the step by step the synthesis of the basic molecule that was required for the synthesis of the pyrimidine nucleotide. So that process or phenomena or pathway is known as the de novo pathway for the pyrimidine synthesis. So regarding the location for de novo pyrimidine synthesis, the location that was occurs in the cytosol of the all type of tissues. Okay, but the purine nucleotides, so the de novo synthesis is mainly occurs in the liver, but small amount or traces amount that will occur in all tissues, but frequently or majorly, so that was occurs in the liver. But here the de novo pathway for the pyrimidine biosynthesis, so that was mainly occurs in the cytosol of the cell of all type of the tissue, means throughout the body. So everywhere the pyrimidine bases are synthesis or de novo pathway can be occurs or happens. So regarding the substrate for the pyrimidine synthesis is carbon dioxide. It is required for carbon dioxide is also donate the carbon atom. So when it is reacts with the water, responsible for the formations of the bicarbonate. So they can donate their own carbon atoms. So then glutamine. So glutamines, so they are mainly donate the amino groups or amido groups. Then ATP, they supply the phosphate. Then aspartate is also same regarding the glutamine, and they are also require the water, NAD, then the phosphoribosyl pyrophosphate, that is PRPP. So these are the substrate. So that was required for the pyrimidine synthesis through a de novo pathway. So regarding the end products after the de novo pyrimidine pathway, that was the UTP, that is uridine triphosphate, that is uracil triphosphate, then CTP, cytosine triphosphate, then glutamate, NADH, and as well as the carbon dioxide. So these are the end products of the de novo pathway or de novo pathway for the pyrimidine synthesis. So regarding the basic molecule, so this is the basic molecule of pyrimidines. So that was required for the synthesis of further pyrimidine nucleotides. Okay, so in this basic molecule, so N1, that is the nitrogen 1 in the position 1, then carbon dioxide at the position 6, that is the sixth carbon dioxide, then fifth carbon dioxide, and fourth carbon dioxide. Okay, so then there is a one, that was nitrogen in the position number 1. So these molecule in the basic molecule is formed by the contributions of the aspartate. So aspartate is an amino acid. So that aspartate can contribute the to add the nitrogen in position 1 and carbon dioxide in the position 4, 5 and 6. Okay, so in the pyrimidine base. Then regarding the N3, that is the nitrogen in the position 3, so that was contributed by a glutamines. And regarding the carbon dioxide in the position 2, so that was contributed by the bicarbonate. So this carbon dioxide, it was reacts with the water in the respiratory systems or in the lungs. So that was counted into bicarbonate. So that bicarbonate is a source for the carbon atom for both purine basis synthesis and as well as the pyrimidine basis synthesis. From this basic molecule, further the UTP, CTP, they are TTP, so they are synthesized. So like that, the pyrimidine, so they are synthesized with the help of the aspartate, glutamate and bicarb. Only three molecules are required. So that's why it is the less complicated. So as compared to the purine synthesis. So now we can learn step by step. So the do you know a synthesis of first UMP, that is uridine monophosphate, from which only the further nucleotides, so they are formed. So UMP, that is uridine monophosphate, is also acts as the precursor for the cytosine monophosphate, and the synthesis of the uracil, that is uridine monophosphate, so that was involved or completed in the 6 steps. The first steps, that is the synthesis of the carbonyl phosphate. So the carbonyl phosphate, so that was synthesized from the glutamine and carbon dioxide. So in the presence of the enzyme, so that enzyme was known as the carbonyl phosphate synthetase 2 enzyme. So this enzyme is only found in the cytosol. Already you learned from the nucleic acid, that is the protein metabolisms in the urea cycle, I think. Okay, so this carbonyl phosphate synthetase 2, it present in the cytosol. It can utilize the carbon dioxide and glutamine, and also it can require the 2 molecule of ATP and as well as water, and responsible for the formations of the carbonyl phosphate. So dear students, the carbonyl phosphate that was formed from the carbon dioxide and glutamine, it was going through the 2 reactions. So in the reaction 1, so that the carbon dioxide reacts with water, it can form the bicarbonate. So that bicarbonate, it can reacts with or combines with the ATP, then can form the carboxyphosphate. So here the ATP, it is a donor for the phosphate. It can donate the phosphate and convert it into ADP. It can loses one phosphate group. So that phosphate group can combine with the bicarbonate and form the carboxyphosphate. And this carboxyphosphate, it can react with the ammonia, and the source of ammonia is, that is the glutamines. So glutamine is the amino acids. Okay, so that can use the ammonia by deaminations process. So that ammonia is reacts with the carboxyphosphate, then forms the carboxylic acid. So like that, in this reaction, in the first reaction that was involved in the formations of the carboxylic acid by use of bicarbonate, ATP and as well as the ammonia. In the reaction 2, inside this reaction, in the step 1, so that is the formations of the carboxyl phosphate. So this carboxylic acid, it can react with the another molecule of ATP. So another ATP, it can supply the another molecule of phosphate, then responsible for the formations of the carboxyl phosphate. So like that, the carboxyl phosphate can formed in the step 1. The step 2, that is the synthesis of the carboxyl aspartate. So carboxyl phosphate, so can converted into, that is the carboxyl aspartate by the presence of the enzyme aspartate transcarbamylase enzyme. So this aspartate transcarbamyl enzyme can convert carboxyl phosphate into a carboxyl aspartate by utilizations of the aspartate. So here the ring is not closed. The ring is formed definitely, but the ring is not closed. So that ring can be closed in the step 3. So that's why the step 3 is ring closure and as well as dehydroorate formations. So carboxyl phosphate in the presence of the, that is the dehydrogenase enzyme, it can undergo the condensation reactions or eliminations reactions through which one molecule of water can be eliminated in the presence of the dihydroorate enzyme. Then the in the carboxyl aspartate, the ring can be closed. So then it was responsible for the formations of the dihydroorate. So in the reaction 2, So one molecule of water is eliminated. So that eliminations of water molecule is helpful for the ring closure in the by the enzyme, that is the dihydroorate. So in the step 4, that is known as the oxidations of the dihydroorate. So here the dihydroorate can converted or oxidized into a orotate. So that oxidation reaction was regulated by NAD. So NADS, it can be oxidized. Okay, it can use the hydrogen ion due to oxidation in the presence of enzyme. So dihydrotate dehydrogenase enzyme. So that enzyme can oxidize the dihydroorate by utilization of the NAD. So here proton donor is a NAD. So then NAD become NADH plus. Then the responsible for the formations of the orotate from the dihydroorate by utilizations of the NAD. So that's why in the step 4, the oxidations of the dihydroorate can be occurs and forms the orotate. Then in the fifth reaction or step 5, that is the acquisitions of ribose phosphate moiety. Okay, that is the acquisitions of the ribose phosphate moiety. So orotate can occupy the one molecule of ribose 5 phosphate from the PRPP. So in the presence of the orotate phosphoribosyl transferase, then it can form the orotatidine monophosphate, that is the OMP. So here the orotate, it can react with the PRPP, that is the 5 ribosyl 1 pyrophosphate. Then it can respond for the productions of the OMP. Step 6, that is the decarboxylations, okay, to form a, that is the UMP. So OMP, it can undergoes the decarboxylations in the presence of the enzyme OMP decarboxylase, and it can release the carbon dioxide. Okay, so due to release of carbon dioxide in the presence of decarboxylase enzyme by decarboxyl reaction from OMP and yield a uridine monophosphate, that is the UMP. So and UMP is the main molecule or basic molecule that was required for the formations of the further pyrimidine nucleotide. Okay, so now we learn regarding the synthesis of the UTP, CTP and as well as DTMP. So this DTMP synthesis will be learned in the next steps. Okay, so that UTP, it was formed by a 3 enzymes. Okay, that is a kinase enzyme. It in the presence of ATP. So that kinase enzyme, it can convert UMP, that is uridine monophosphate into a uridine diphosphate. It means consist of the 2 phosphate. So that phosphate can donated by the ATP. So that's why. So this reaction is required the ATP. So then the ATP can loses the one phosphate group and it becomes the ADP. So then that phosphate group can react with the uridine monophosphate. It become the uridine diphosphate. Then presence of the further the enzyme kinase, the same enzyme, it required another molecule of ATP. Then it convert UDP into UTP, that is uridine triphosphate. So that uridine triphosphate is the basic molecule for synthesis of the ribonucleotide. Okay, that is required for the synthesis of the base pair in the ribosomes. So those, that's why they are known as the ribose nucleotide. So that was found from the uridine monophosphate in the presence of the 2 molecule of ATP and the kinase enzyme. So like that the uridine triphosphate was formed. Then further the UTP, that is uridine triphosphate. So in the presence of CTP synthetase enzyme, it was converted into, that is a cytosine triphosphate by presence of the glutamine, ATP and water. Okay, glutamine, ATP and water. Glutamine supply the amide group, ATP supply the phosphate group and water is required for the hydrolysis. Okay, so these water and glutamine, ATP, so they react with the uridine triphosphate in the presence of the CTP synthetase enzyme. Then CTP will be the form. So like that UTP, CTP, so they are formed in the from the, that is the uridine monophosphate by presence of the kinase enzyme as well as synthesis enzymes. And this UTP and CTP, so they are involved in the formations of the base pair in the ribosomes. In the next step, we can study regarding the synthesis of deoxyribonucleotides, because the for ribonucleotides, so they required for only the base formations in the RNA. For DNA base pair, we require the deoxyribonucleotides. Okay, so that's why the deoxyribonucleotides, so they are also formed from the ribonucleotides. That was from the RNA. Okay, so the from the RNA, so what are the base pairs are there required for the RNA synthesis? So they are formed by a de novo pathway or as well as the cleavage pathway. So then those ribonucleotides, so they are involved in the formations of the base pair in the RNA. Same ribonucleotides, so they are also required for the formation or conversions of the deoxyribonucleotides, and these deoxyribonucleotides, so they are required for the formations of the base pair in the DNA. So this reaction was catalyzed by a one enzyme. So that enzyme is known as a ribonucleotide reductase, that is the RNR. So this enzyme, it was reducted in the position carbon atom 2 in the ribonucleotides. So this enzyme, it can cause the reduction in the position C2. Okay, so that is it attached with the OH group. Then it can converted into a deoxyribonucleotides, and this enzyme, it is categorized into three classes. That is the class 1, class 2, class 3. So all the enzymes rule is same, but they are differ in the prosthetic group. So like that, in the ribonucleotide, the OH was replaced by a H hydrogen in the presence of the ribonucleotide reductase enzyme. Then the ribonucleotide become the deoxyribonucleotides. So that deoxyribonucleotides, so they are involved in the formations of the base pair in the DNA. Okay, so that is the overview regarding the synthesis of the deoxyribonucleotides from the ribonucleotides. So these are the ribonucleotide, that is the adenine diphosphate, guanine diphosphate, cytosine diphosphate, then uracil diphosphate. Okay, so the enzyme, that is the ribonucleotide reductase, it can convert ADP into DADP, deoxy adenine diphosphate, then GDP into deoxy GDP, then CDP into deoxy CDP, then UDP, the uracil diphosphate is converted into that is deoxyuryl diphosphate, present same enzyme. And further, in the presence of the thymidine synthetase enzyme, so that can convert DUDP into that is uracil monophosphate, because for DNA we require the thymine pyrophosphate, not a uracil pyrophosphate. So that's why, so in the presence of this enzyme, the thymidine synthetase, it can convert UDP into a TDDP, that is deoxy, that is a thymine diphosphate, in the presence of the thymidine synthesis enzyme. Further, it can TMP, then DTDP by the enzyme this. Then presence of the another enzyme, that is the DNTP kinase, it can convert all deoxy diphosphate into a triphosphate. That is the DADP is converted into TADP, the deoxyadenosine triphosphate, in the presence of the DNTP kinase. And so like that, that is the DGTP, deoxy-GTP, deoxy-CTP, and deoxy-TTP are formed. So like that, the deoxyribonucleotides, so they are formed from the ribonucleotides. nucleotides in the presence of the ribonucleotide reductase enzyme as well as the DNTP kinase enzyme, then as well as thymidine synthase enzyme. So then they become the deoxyribonucleotides, and these are utilized in the synthesis of the DNA by an enzyme that was known as the DNA polymerase enzyme. So this enzyme is involved in the sequencing in the DNA pair. So like that, so these are enzymes. If any micro in the microsome also the same process occurs. If any drug is inhibit any one enzyme means, so that drug will be show helpful for treatment of the cancer or helpful for to show the antimicrobial activity. Okay, so next, so we can learn regarding the degradations of the pyrimidine bases. So that is cytosine monophosphate. It can phosphorize, release the phosphate group and become a cytidine. And cytidine further undergo the hydroxylation, then release the ammonia, it becomes the uridine. In another side, the uridine monophosphate, it is also undergo the phosphorylations, that is the dephosphorylation, that can release the phosphate groups, then it becomes the uridine. And further uridine, it can undergo the phosphorylation and lose the ribose 1 phosphate and it become the uracils. So uracil in the presence of the NADPH and plus H, it can form the NADP, then it becomes the dihydrouracils. Further additions of water, so then it can form the uridopropionate. So then 1 mole of additions of water, so then the uridopropionate is converted into beta alanine. So in the presence of alpha ketoglutarate, so it is the intermediate compound for the transamination reactions. That is alpha ketoglutarate is also required here. That is the catabolism of the pyrimidine bases. Okay, so the beta alanine in the presence of the alpha ketoglutarate, then alpha ketoglutarate is converted into glutamate, and the beta alanine it becomes the malonate semi-aldehyde. So in the presence of the coenzyme A and as well as NAD, it means it can undergo the oxidation reactions. That is the malonate semi-aldehyde. It can undergo the oxidation reactions in the presence of NAD and as well as the coenzyme. Then it can form the acetyl CoA. Then the NAD converted into NADH. Then it can undergo the electron transport chain. So where it can utilize, release the ATP, that is about 2.5 ATP. Then the coenzyme is converted into carbon dioxide. It releases the carbon dioxide and some enzyme CoA part that can bind in here, it becomes the acetyl CoA. And this acetyl CoA undergo the beta oxidation and it can release the energy, are helpful for the release of the energy. So like that the pyrimidine is also involved in the formations of the ATP RNA. So this is the regarding the catabolism or degradations of the pyrimidine bases. So next, so regarding the important enzymes and the regulations, so the pyrimidine biosynthesis that was occurs in the presence of the main enzymes like a carbyl phosphatase synthetase 2, then as well as the uridyl OMP decarboxylase and as well as the TP synthetase enzyme. So these are three main enzymes. So that was involved in the or they are regulated the that is the de novo synthesis of the pyrimidine bases, and the carbyl phosphate synthetase that was inhibited by the, that is the UTP and as well as activated ATP and as well as PRPPs. If their concentration is more means automatically the de novo synthesis of the pyrimidine bases that was stopped or inhibited. And orotin decomposes enzyme activity that was inhibited by the UMP as well as the CMP, the UDP, uridine monophosphate and cytosine monophosphate. Then TP synthetase enzyme that was inhibited only by the CTP, that is cytosine triphosphate. So like that, so these are the various component are enzymes. So they are regulate the de novos in the either they stimulate or inhibits. Okay, so next we can concentrate regarding the pyrimidine synthesis via slavage pathway. So that is the pyrimidine slaved from the ortic acid or uracil and thymine, but not from the cytosine. So the pyrimidine, so they are reutilized, okay, by a slavage pathway. Whatever the metabolites are there from the pyrimidine catabolism like a arotic acid, uracil and thymine. So they are utilized for the formations of the pyrimidine bases, but the cytosine it cannot be reutilized. So that's why we are suffering from the cystinuria due to because it is not utilized. Okay, then the slavage pathway, so that was completed by the enzyme. That enzyme is known as the pyrimidine phosphoribosyl transferase enzyme. So this enzyme it can utilize the orotic acid, uracil and thymine and involved in the synthesis of the pyrimidine bases, and that pathway is known as the slavage pathway. Okay, so next we can learn regarding the disease related to the abnormal purine and pyrimidine metabolisms. So now we learn regarding the disease related to the abnormal purine as well as the pyrimidine metabolisms. So between purine and pyrimidine metabolisms, so diseases are more commonly seriously caused due to only purine metabolisms, that is abnormal metabolisms, not related to that much of the pyrimidine metabolisms. So these are the important disorders or main disorders I am here highlighted. Okay, so that is related to purine. There is a block of the degradations occurs with the syndromes involving if any blockage in the malfunctions or mutations in the enzymes, so that was involved in the degradations of the purine, so that was responsible for to cause of the, so these main diseases, that is immune deficiency, the severe deficiency of the immunity due to lack of degradations of the purine, because they are inhibit the formations of the immune cells like a T lymphocytes and B lymphocyte. Due to lack of the purine degradation, our immune cells cannot be multiplied. So due to that, the person was suffering from the severe immune deficiency, as well as the peoples, so they are suffering from the, that is the myopathy, even the blockade of the degradations of the purine, so that were responsible for the development of the renal calculi. Okay, and regarding the increase the degradations of nucleotides, that is also occurs some syndromes. So those are hyperuricemia. So more degraded means more uric acid is formed, because the end product of that is the purine nucleotide is a uric acid. So that's why if it is more degraded means the hyperuremia occurs or hyperuric acid is occurs in the plasma. So that uric acid was accumulated in the joint. So due to that, it was responsible for to cause of the inflammation of the joint or blockage of the joint. So that condition is known as the gout. And as the uric acid is increases in the kidney, so those uric acid was counted into a kidney stone. So that's why, so those are responsible for to cause of the renal calculi. And also the person was suffering from the anemia or acute hypoxia due to increase the degradations of the that is purine nucleotides. So regarding the pyrimidines, the person is suffering from more commonly, that is the aciduria. So they due to accumulations of the ortic acid. So that was found in the urine due to abnormal metabolism of the pyrimidines or catabolism of pyrimidines. So that condition is known as the aciduria. So these are the important disorder related to the purine and pyrimidine metabolisms. Okay, first when we concentrate regarding the hyperuricemia, it is characterized by plasma urate or uric acid level is greater than the 7 mg per deciliter. Okay, due to over degradations of the purine bases, the uric acid will be more formed. So that was characterized by increase in the plasma concentrations of uric acid more than 7 mg per deciliter. So that condition is known as the hyperuricemia. So normal levels of the plasma level of uric acid is in female 2.4 to 6 mg per deciliter, in the male 3.4 to 7 mg per deciliter. If it is beyond that, so that condition is known as the hyperuricemia. So once again, the hyperuricemia, so there are two types. That was the primary hyperuricemia and secondary hyperuricemia. Primary hyperuricemia means an intake defects in the purine metabolisms, or there is a decrease in the uric acid excretion. Any defects in the purine metabolism that can enhance the purine metabolisms, or there is a decrease in the urine excretions or uric acid excretion through urine. So that condition is responsible for to elevate the plasma uric acid level. So that type of hyperuricemia is known as the primary uricemia. So with the help of the diet or with the help of the diuretics, we can control the treat the primary hyperuricemia. And secondary hyperuricemia, that is the increased availability of the purines due to medications or medical conditions or through a diet. If you are consuming more nucleic acid containing diet for the sake of to gain the body weight or as a protein supplements or to build up the mass, the purpose of those people they are joining the gym, so they prefer the more protein food or more nucleic acid to build up the more mass. So that condition of diet or use of certain medications or medical conditions like diseases, so those are responsible for to increase the uric acid level in the blood. So that condition is known as the secondary hyperuricemia. So next one, so that is regarding the gout. So gout, it is the inflammatory disease or disorder related to the joint or the accumulations of the, that is the sodium urate crystal in the joint. Okay, so that condition is known as the gout. So due to accumulations of sodium urate crystal in the joint, that was responsible to cause of the inflammation and pain. So due to inflammation, pain, if you are not treated, if you are not corrected your uric acid level in the blood in the joint with the help of the medication, then that joint was blocked, and due to blockage of joint, the movement is not possible. What are locomotions in your grandma, grandfathers? So their joint is blocked because of the urine, that is the accumulations of sodium urate crystals in the joints. Due to that, so they are unable to move. And if you want to detect the gout in early stage and need to, you want to give the treatment to the your father, mother or grandfather and grandmothers in early stage, simply you can watch the thumb of the both leg, because so as the crystal our uric acid crystal is start to deposit in the joint, in the knee joint as well as the ankle joints, the first, so they are causing the sign and symptoms regarding the swelling of the thumb in the leg. If the leg was more swell in the leg, the thumb is more swell in the leg, that indicates so that candidate was suffering from the gout in the early stage. You can treat that. In early stage, you can advise the medications or food or control on the diet, so that was decrease the degradations of the purines. Okay, or you can enhance the syllabus pathway through a aerobic exercise or meditations or certain vegetables or antioxidants. So then, so our parents or grandmothers, grandfathers, they are free from the gout by simply by absorbing the, so their leg thumb. So that was a more swell, that indicates those candidates they are suffering from the gout. If you are not corrected, not consult with the doctors or not take any medications or therapy, definitely after 1 year or 2 year, so their knee all joints are blocked. So due to blockage of joint, so they are unable to walk, sit wherever. Okay, so there all locomotor activities can be decreases. So once again they create the problem to your family members and yourself. So that's why in the early stage you can detect and give the therapy. So with the help of the supervisions of the doctors, or if you have any herbal drug, so then you can give the herbal drugs daily, because they are the most safe as compared to the allopathics. Okay, so regarding the symptoms in the affected joints, so they are pain, then swelling, redness and heat is increases. So these are the early signs and symptoms of the gout. regarding the causes. So there is a problem in the uric acid excretion because they are holding the more urine, so they do not empty the bladder. So when they fill the urine, that was also one of the problem risk factor for to cause of the gout. So that's why the gout is more problem in the female as compared to the male. Okay, so that is one of the one reason. So to avoiding the micturations or to avoiding the gas, that is a bladder empty. Then second, the diet which can contain rich of purines or alcohol are deficient in the dairy products. So that was also responsible for to cause of the gout. So another, to increase the purine degradation due to exposure to the sunlight, UV rays, or many other certain chemicals, or by eating of the more pesticides food, imported food, okay, imported chocolates, imported ice creams, we are preferring the pizza vagera vagera, so they are containing certain chemicals. So those chemicals, so they are specially enhance the degradations of the more and more and more purines. So that's why as compared to rural people, okay, the city people, okay, so they are more risk factor for gout due to only, so they are preferring the imported or chemical mixed food. Okay, that is one of the reasons for to increase the purine degradation and responsible to cause of the gout. So other, to increase the PRP synthetase activity, that is also one of the risk factors for gout, because so that enzyme is increases the productions of PRPP, and also it can increases the purine synthesis because it is also basic molecule required for the purine as well as the pyrimidine synthesis. Then the synthesis increases, then as the synthesis increases, the degradation is also increases. So as the degradation increases, there is increase in the uric acid production, so that was accumulated, then it can cause the gout. Then the decrease the partial HGPRT. Okay, so this enzyme, it was required for the D, that is the Slavase pathway synthesis of the purine. A deficient of this enzyme, then also the de novo synthesis is more occurs, so that can accelerated. So due to that, more metabolized and more uric acid will be produced. So that's why this enzyme is also responsible cause of the gout. So for the treatment of gout, we are simply using the non-steroidal anti-inflammatory drug like a paracetamol, mefenamic acid, ibuprofen. So any painkiller drug belongs to non-steroidal anti-inflammatory drugs. So they are helpful for treatment of the gout. Our main standard drug, that is the colchicine. The colchicine is the one of the best drug or choice of the drug used for treatment of gout, and as well as allopurinol. It is the another drug, so that is also used for the treatment of the gout. But if you are adjusting the biochemical reaction through a diet or therapy, then also you can avoid the gout. Next one is the kidney stones. It is also one of the disorder related to the increase or decrease degradations of the purine metabolism. So in which the uric acid present in high concentration in the blood. Okay, it may be participate in the salt formation in the kidney. So that salt responsible for the formations of the kidney stones, or it can cause the pain, infection, or kidney damage due to more uric acid in the blood that can convert the salt in the kidney. So when the blood visited to the kidney, so then the kidney was suffering from the kidney stone or damage. So that's the kidney stone is another problem related to the purine metabolism. So next one is the Lesch-Nyman syndrome. It is a genetic disorder that was occurred due to deficiency of the this HGPRT enzyme, that is the hypoxanthine guanine phosphoribosyl transferase enzyme. So this enzyme is required for the synthesis of the purine by Slavase pathway. Okay, so as the Slavase pathway is deficient, automatically the de novo synthesis of purine is increases. Automatically the purine degradation is increases. Automatically the uric acid level is increases due to problem in the Slavase pathway by deficiency of this enzyme due to genetic mutation, because it is a genetic disorder from birth it was present, and that condition is known as the Lesch-Nyman syndrome, that is LNS. So it is present at the birth in the baby boys. So once again, it was present in the boys, not in the girls. Okay. So hypoxanthine and guanines are not used in the salivary pathway for purine nuclear synthesis. That was another reason for to cause of the LNS. Okay, if they are not utilized, then they are converted into a uric acid in the final step. So then hypoxanthine is converted into xanthine. Then xanthine is converted into uric acid. If they are not utilized, and guanine is converted into hypoxanthine, then hypoxanthine into uric acid. If both are not utilized by salivary pathway by deficiency of this enzyme, then the uric acid production is more. So that condition is known as the Lex-Nehman syndrome or LNS. Symptoms regarding this disease: LNS, severe gout, severe mental and physical problems, and self-mutilating behavior. So these are the various symptoms that was related to the Leishman that was early detected in the baby boys at the birth. So last one, that is the severe combined immunodeficiency, also written as the SCID. Okay, it is occurs due to the decrease in the degradations of the purines. Okay, that is the adenosine deaminase deficiency that was required for the degradations of purine. Due to that, there is no degradation, and due to that, it can cause the accumulations of the DATP. Then this DATP, it can inhibit the ribonucleotide reductase. Then also it can inhibit the multiplications of the B and T cells. So they are unable to fail due to inhibitions of ribonucleotide reductase or accumulations of the DATP. So then severely our immunity can be decreases. So that condition is known as a severe combined immune deficiency or SCID. So next one is that is the last condition. So that was related to the pyrimidine disorder, that was known as the aorta aciduria. It is also inherited disorder of pyrimidine synthesis. So that was caused by deficiency of enzyme, that is aortate phosphoribosyl transferase and as well as the decarboxylase enzyme. So these are involved in the mainly the metabolism of the pyrimidine. So due to deficiency of this enzyme, so there is an abnormal in the metabolism of the pyrimidine. Then the person was suffering from the high concentrations of the aortate in the urea. So that condition is known as the aortic acid urea. Symptoms: the excess of aortic acid and it excretion in the urine about 1 to 1.5 gram. Then as well as there is a mental and physical retardation is there. Then also the person was suffering from the megaloblastic anemia. So these are the signs and symptoms of the there is aortic acid urea. Okay, so these are the various disorder related to the purine and pyrimidine metabolisms.